Imagine a vast library filled with countless books. Each book holds the instructions to build and operate a living organism. These instructions are stored in our DNA, the blueprint of life. DNA is made up of genes, which are like individual chapters in these books. Each gene carries a specific set of instructions, determining traits like eye color, height, and even susceptibility to certain diseases. These instructions are written in a chemical language using 4 letters, A, T, C, and G. These letters represent the building blocks of DNA called nucleotides. The specific sequence of these letters determines the information encoded within a gene. Any alteration in this sequence, whether a single letter change or a larger rearrangement, is called a genetic mutation. Genetic mutations are the source of all genetic diversity. Think of mutations as typos that arise when DNA is copied during cell division. Most of the time, these typos are minor and don't have any significant effects. However, occasionally a mutation can alter a gene's instructions in a way that changes a trait. These changes can be as subtle as a slight variation in hair color or as dramatic as a difference in disease susceptibility. Over time, these small changes accumulate within a population, leading to a wide array of variations. This diversity is essential for the survival and adaptation of species. While we often think of mutations as harmful, many mutations are actually beneficial. These mutations can provide an advantage to an organism, making it better suited to its environment. For instance, a mutation might allow a plant to withstand drought conditions better or help an animal develop camouflage to evade predators. These advantageous mutations are more likely to be passed down to future generations because individuals carrying these mutations are more likely to survive and reproduce. Over long periods, these beneficial mutations can accumulate, leading to the evolution of new species. Section 4. When genes go wrong, mutations and disease. Not all mutations are beneficial. Some mutations can disrupt the normal function of genes, leading to diseases. These disease-causing mutations can be inherited from parents or arise spontaneously during an individual's lifetime. Some diseases, like cystic fibrosis and sickle cell anemia, are caused by mutations in single genes. Other diseases, such as cancer, are more complex and can be caused by mutations in multiple genes, often in combination with environmental factors. Section 5. Examples of mutations in action. One striking example of a beneficial mutation is the development of lactose tolerance in humans. Ancestral humans could only digest lactose, the sugar in milk, during infancy. However, a mutation arose that allowed some individuals to continue producing the enzyme lactase into adulthood. This mutation provided a nutritional advantage, particularly in societies that domesticated cattle. Consequently, this mutation spread rapidly through these populations. On the other hand, the mutation that causes sickle cell anemia provides an example of how a single mutation can have both beneficial and detrimental effects. This mutation alters the shape of red blood cells, making them inefficient at carrying oxygen and leading to various health problems. However, individuals with one copy of the sickle cell mutation and one normal copy are protected against malaria. Section 6. The duality of mutations. A recap. Genetic mutations are the driving force behind evolution, generating the diversity that allows species to adapt and thrive. While some mutations can be detrimental, leading to diseases, others can be beneficial, providing advantages that fuel the process of natural selection. Understanding the dual nature of mutations is crucial for comprehending the complexity of life on Earth, from the evolution of new species to the development of diseases. Our genes tell a story, a narrative of change and adaptation written in the language of DNA.